This table is comparing graded potentials and action potentials. So you should be able to compare and contrast them. Remember, graded potentials are usually in the dendrite or the axon. There's some exceptions to that if it's an axonic synapse, but it's usually in those two areas. The action potential is going to begin at the initial segment, which is also called the axon hillock or the trigger zone. And that's going to be because there's a high concentration of sodium gates. Graded potentials are only going to travel a short distance, typically within the cell body, to the initial segment of the axon, whereas action potential travels a long distance from the trigger zone through the entire length of the axon. And that could be something as long as a sciatic nerve, which goes from your hip all the way down to your foot. The amplitude or the size varies in graded potentials but decays with distance, whereas in action potentials, they are all the same, which means all or none. So they do not decay with distance. The stimulus for opening the ion channel in graded potentials is a chemical like a neurotransmitter, or there could be a sensory stimulus like light pressure or temperature, producing a graded potential. In action potentials, it's a voltage gated channel. So there's depolarization, and it could be triggered by multiple graded potentials, which we're going to see coming up, which is the concept of summation. There's an positive feedback cycle in action potentials because the same thing happens over and over. So once we have one action potential at a node of Ranvier, then we have another action potential at a node of Ranvier, but we don't have that in graded potentials. For repolarization, it's voltage independent, and it occurs when the stimulus is no longer present. But in action potentials, it's voltage regulated. So it occurs when sodium channels are inactive and potassium channels open. For summation, the stimulus responses can summate to increase the amplitude of a graded potential. So if we were to look at a graph here, and we saw, let's say that all four of these were excitatory. So that means that they all depolarize the membrane. So we would have threshold here at negative 55. It begins at negative 70. We have one excitatory postsynaptic potential that depolarizes, another after that that depolarizes, another after that. And once we reach negative 55, we then automatically have an action potential. So it goes up like this and then back down. So that's what summation means. If it's summated in a time dependent fashion, meaning they're right after each other, that's called temporal. But if they come from different axons, it's called summation. If this is an excitatory, this is an inhibitory, and this is an excitatory, if we were to look at a graph as well, with our two magical numbers of negative 70 as the resting membrane potential, negative 55 as the threshold, we would see, let's say that we have an excitatory one that goes like this. Then we have an inhibitory one that goes like this. Then an excitatory one that goes like this. You can see that there's really there's still kind of summation, but they basically cancel each other out. So it never reaches that magical number of negative 55 for the threshold. Now, occurring at the synapse, there is what we call synaptic delay, and this means that the neurotransmitter must be released. It must diffuse across the synapse and bind to the receptor. So there's a time delay of about 0.3 to 5 milliseconds, and it's a rate limiting step of neural transmission. So we're going to continue comparing graded potentials and action potentials. And I'd like to encourage you to go to this figure animation in Mastering A&P as well. As you know, in the study area, there's lots of different animations that can help you. So the function of a graded potential is, again, it's a short distance. When it's summated, it determines whether an action potential is generated, whereas action potentials are long distances and they're referred to as a nerve impulse. When we use postsynaptic potential, it's only referring to graded potentials. If it's excitatory, it depolarizes, spreads to the initial segment of the axon, and it increases the likelihood of an action potential. If it's inhibitory, though, it hyperpolarizes the membrane and decreases the likelihood of an action potential. So two examples of these are EPSPs, open chemically gated channels that allow both sodium and potassium to move at the same time. This is different than action potentials. Remember, action potentials has a separate sodium gate and a separate potassium gate. In an EPSP, we have both sodium and potassium moving at the same time. This is why it's only a slight sub-threshold depolarization. If it's an IPSP, an inhibitory postsynaptic potential, it opens chemically gated potassium channels or chlorine gated channels. And we have to think of the net result. So the inside is going to become more negative. That's what hyperpolarization means. Whereas depolarization means that it becomes more positive. So in the example of the EPSP, if we have a cation that enters, that makes it more positive. Or if we have a cation leaving, that makes it more positive as well. In the IPSP example, if we have a potassium that's leaving, it makes the inside more negative. Or if we have an anion that's entering, it also makes the inside more negative. Whereas in the action potential, remember, we have voltage gated channels. We have a sodium channel, and then we have a potassium channel. So sodium enters, goes from plus 30, it goes from negative 70 to plus 30. And when potassium leaves, it goes from plus 30 down to negative 90 with the potassium leaving. The peak membrane potential here is if we have an EPSP, it moves towards zero because it starts to become more positive or less negative, starts to move towards threshold. For an IPSP, it hyperpolarizes, so it becomes more negative on the inside. For the action potential, this is what we see here, and it can range in amplitude. So going from negative 70 to plus 30 is quite a large amount. Going from negative 70 to plus 30. And we're just going to kind of ignore this part so you can cross it out.